Hello everyone, you are all welcome to lecture number 1234 and that is in continuation of cholesterol chemistry. Now I am going to discuss the purification of cholesterol because when we are getting steroid from any natural source, it is having lot of impurities and we have to purify it. There are several methods, but the best known method to purify the cholesterol from impure steroid, that is known as bromination and debromination of steroid or cholesterol. So what we are doing first, we will get the impure cholesterol and it is subjected to bromination. We will get dibromo derivative of cholesterol, and that dibromo derivative of the cholesterol is again subjected to zinc dust distillation. So zinc dust will remove the bromine and 100% pure precipitate of cholesterol will be formed, and the zinc bromide, that is ZnBr2, will go in the solution in the form of impurities, and we can filter the 100% pure precipitate of cholesterol. In this way, the cholesterol is 100% purified from impure steroids. Now the second reaction, I have already told you that sometimes I may ask the question. The students should remember because this lecture is for steroidal chemistry. How will you prove the presence of alcoholic group in steroid, and how will you prove that alcoholic group is acid stable? So in that case, you have to discuss two reactions. First reaction, which I have already discussed, that is Jones oxidation. You will get keto form, keto derivative of cholesterol, and that clearly indicates that the hydroxy group means alcoholic group in cholesterol is acid stable, because those compounds which are acid stable can undergo Jones oxidation. Secondly, when cholesterol is subjected to acetylation in presence of pyridine, because pyridine is used as a solvent, and that pyridine plays very important role as a solvent to produce acetylonium ion, and their reaction mechanism is there. But here we are not interested in much reaction mechanism of cholesterol, rather how to get the ester. So once it is subjected to in acetic anhydride in presence of pyridine, it is converted into ester, that in the form of acetate, and the conversion of cholesterol into ester in the form of acetate confirm that cholesterol contains alcoholic group, hydroxy group, because if it is not alcoholic, the formation of ester is not possible. So these are all reactions. It is written here, cholesterol. This cholesterol, when it is subjected to bromination, you are going to get dibromo cholesterol, and impurities will be there in solution. Then it is again distilled with zinc dust. When it is distilled with zinc dust, you will get pure cholesterol melting point 146 degrees centigrade temperature, and all the impurities including with ZnBr2 will be in solution. In this way you can get the pure cholesterol. The second is about alcoholic group in cholesterol. When cholesterol is subjected to acetylation, acetylation means when it is treated with acetic anhydride in presence of pyridine, it is converted into acetylated cholesterol, and that acetylated cholesterol is nothing but it is the ester derivative of cholesterol, and the conversion of cholesterol into ester derivative confirms that cholesterol contains alcoholic group. So here is summary is given here. See definition, what is steroids? The steroids form a group of structurally related compounds and which are widely distributed in animal and plant kingdom. Number two, the word steroid is derived from sterol, and that is meaning vitamin D and also bile acid. The nucleus and structure of steroid is based on 1,2-cyclopentanophranthine nucleus, and that is here phenanthrene and cyclopentano. All steroids must give these hydrocarbon. I have already discussed all this, just I have given the note. Hydrocarbon on dehydrogenation when it is distilled with selenium at 360 degree temperature. In fact, steroid could be defined as any compound which yields hydrocarbon, and where it is written the structure. Steroids occur in animals, plants, and fat, oil, and they are crystalline compounds and contain alcoholic group. They occur free as a ester in higher fatty acids, and the steroids obtained from animal is called zoosterol. The one which is obtained from plant source is called phytosterol, and the one which is obtained from yeast or fungi is called mycosterol. And here there are some very important two tests to know the steroidal nucleus, whether it is present or not. So first we have to do the color reaction test, and the most important reaction one is called that is Lieberman-Burkhardt reaction. I will give the ratio, and the second one is called Slavovsky test. So these two tests are there which will confirm whether that particular compound is steroid or not. So now I am giving the summary of the same what I have already discussed in lecture, that is cholesterol. So what is cholesterol? This is the sterol of higher animal. It is the main product of cholesterol. If we are saying cholesterol, it is animal product, because there are a lot of vegetable oil people, they write cholesterol free. It is rubbish, because cholesterol has nothing to do with vegetable oil. It is always cholesterol free. Cholesterol is animal product of higher animals, occurring free in the combined state in the form of esters. It was first isolated from human gallstone. It is almost the full cholesterol. So human gallstone is nothing but it is cholesterol. The main source of cholesterol are fish liver, human brain, and spinal cord of cattle. The word cholesterol is derived from Greek, means chol means bile. So whole bile is nothing but it is cholesterol. So the word cholesterol came from Greek terminology. The solution, so the compound of bile duct is cholesterol. Cholesterol is white, crystalline, solid, optically active, and gives some color reactions. And these two color reactions I have given you. First, that is called Lieberman-Burkhardt reaction, and in this reaction you will get green color once the cholesterol solution in chloroform, where acetic anhydride and cyanide tube concentrated sulfuric acid is added. Immediately it will appear into green color, that confirms it contains cholesterol. The second is Slavovsky test. When concentrated sulfuric acid is added to the solution of cholesterol, a red precipitate. The formation of red precipitate in presence of concentrated sulfuric acid confirms that this substance is purely cholesterol. So with this I will stop, and I will continue in the next lecture, where I will be discussing some important hormones. They are known as endocrine hormones. They play a very important role in physiological activity of animals. Once again, thank you very much for watching the lecture.